Welcome viewers to another exciting journey of learning zoology with me, Arunima. In this session, we will be discussing the process of metabolism of carbohydrates, or in other words, we will learn how the carbs that we consume daily gets utilized in making the energy packets for our each and every cellular functions. It is the ATP molecule that is produced and which provides us the necessary energy needed to carry out daily activities. The carbohydrates that are a part of our daily diet gets digested by certain intricate physiological processes, and the simpler molecules of glucose are produced, which are taken up inside the cell and are utilized in turn to produce energy in form of ATP molecules. Let us now have a quick look at the objectives of our study, which are to learn glycolysis pathway, to learn the Krebs cycle, to learn the processes of glycolysis and gluconeogenesis, to learn about the electron transport chain, and also to learn how ATP is synthesized. Along with it, now let's have a brief idea about the learning outcomes of this topic. By the end of this topic, you will be able to explain how carbohydrates are metabolized and ATP or energy packets are produced for various cellular functions, the anabolic and catabolic processes involved in carbohydrate metabolism as a whole. You will also be able to explain the detailed process of glycolysis, that is the breakdown of glucose or hydrolysis process, and how pyruvate is produced, is the precursor for Krebs cycle. You can therefore be able to describe the pathway of a pyruvate molecule through the Krebs cycle, explain the transport of electrons through the electron transport chain, describe the process of ATP production through oxidative phosphorylation, and also you will learn the anabolic pathway of production of glycogen protein from glucose and also the breakdown of it in times of anaerobic respiration in the process named as glycogenolysis. Here what you can see is a summary of the processes involved in carbohydrate metabolism. Anabolism is a term that defines any process concerned with the synthesis of substances, whereas catabolism refers to the breakdown, and together they are included in the process of metabolism. Glucose is the end product of carbohydrate digestion, which is absorbed into the circulatory fluid, the blood, and is taken up by the cells in order to utilize it in synthesizing ATPs. Glucose is the first phosphorylated in glucose 6-phosphate inside the cells, and conversely, when there is a lack of glucose in blood, the liver cells dephosphorylates the glucose 6-phosphate molecule into glucose and releases them in bloodstream. These G6P molecules are of diverse use. They can be used to synthesize glycogen via glycogenesis in the liver cells, or can be utilized in making pyruvate via glycolysis, or can enter the pentose phosphate pathway to produce 5-carbon sugars and NADPH. Conversely, glucose 6-phosphate can be formed when glycogen is broken down or can be formed from pyruvate too. During anaerobic conditions, when there is an increased demand of ATP and pyruvate is quickly converted into lactate in the muscle, certain amino acids or even glycerol can produce pyruvate that can enter the ATP production cycle. During the aerobic conditions, however, lactate is converted into pyruvate that produces glucose 6-phosphate. There are two pancreatic hormones which play a crucial role in maintaining the balance of glucose in the bloodstream, which is insulin, aiding in glucose uptake, and glucagon, aiding in release of more glucose into the bloodstream. Whenever there is a decrease in the circulatory level of glucose, the pancreas is stimulated to secrete glucagon, which activates the liver cells to metabolize glycogen and release glucose in blood. And if the glucose level increases, then the pancreatic beta cells are stimulated to secrete insulin hormone, which facilitates uptake of glucose in cells and then directs them for the supply of metabolic energy or glycogenesis or fat synthesis, etc. And blood glucose level is restored to normal, and this way the equilibrium is maintained. Let us have a look into the process of glycolysis along with gluconeogenesis. Both the processes share common intermediates and are most abundant processes of carbohydrate metabolism, that is catabolism and anabolism respectively. There are 8 intermediary stages involved, as you can see. In glycolysis, glucose is converted into glucose 6-phosphate by the help of the enzyme hexokinase, and is actually a phosphorylation process where one molecule of ATP is converted into ADP, and the glucose is phosphorylated to form the G6P molecule. G6P is then isomerized into fructose 6-phosphate by the action of the enzyme phosphohexose isomerase. The substrate and the product are interconvertible, and the same enzyme is capable of doing the needful.